RNA purification from blood samples. Usually 1 ml blood can produce 3 microgram of the RNA. While purifying the RNA by using the blood samples, there are a lot of RNases that usually degrade the RNA. So there can be contaminations. All the equipments and all the tubes that are used for purification of RNA while using the blood samples, they should be sterilized and they should be RNase free. Means there should not be any kind of the RNase enzyme that can degrade the RNA. This is the procedure how to purify RNA from the blood samples. The first step is to collect the nuclei or the pellet of the white blood cells. So this procedure describes that how we can collect the nuclei of the white blood cells. So this is the first step for purification of RNA from the blood samples. During this process, we will collect the white blood cells and the nuclei that contain DNA or RNA. So during this step, add fresh human blood in a conical centrifuge tube. Then there is addition of RBC lysis buffer, red blood cells, because the first step while purification of the RNA is to destroy or to lyse the red blood cells, because red blood cells do not contain DNA or RNA. So by adding RBC lysis buffer, the RBC cells, they will lyse, and it should be kept after the addition of the buffer at room temperature for 10 minutes. So there should be a completion of the process, or there should be lysis of the red blood cells. Once they are lysed, then the pellet cells by centrifugation, that can be at approximately 14,000 rpm for 10 minutes. This process that makes the pellet of the red blood cells, because they have already been broken, and the white blood cells, they settle down in the bottom of the tube. In the next step, this process is repeated. So gently resuspend the pellet in the RBC lysis buffer once again, and it is transferred to a small tube, that is 1.5 ml tube, which is also called as microcentrifuge tube. So let's stand for the sample for 2 minutes, and during the next step, pellet cells for 2 minutes after centrifugation in a microcentrifuge, carefully aspirate the supernatant. Process the white blood cells which are now present at the bottom of the tube for RNA desired. If desired, then those white blood cells which are now in the form of the pellet at the bottom of the tube, that can be further processed for the purification of the RNA, or they can be stored at -70 or -80 degree whenever there is need of RNA purification. So during RNA purification, the first step, when we are going to purify RNA from the blood samples, the first step is to prepare the pellet of the nuclei which are present in the white blood cells. So during this lesson, we just have prepared the nuclei which were present in the white blood cells.